I want to talk a little bit about the Hardy Weinberg Principle. The idea here is if Mendelian segregation and random mating are the only processes that affect the gene pool, then the frequency of alleles in a population will remain constant. Ultimately, we're not really interested in the constant maintenance of alleles. We're interested in how genetic frequencies change over time in response to selection or what have you. But first we need to set the baseline or the control, which is how do genetic frequencies behave assuming only Mendelian inheritance and recombination events during meiosis. So this is a problem from your textbook. And I want to briefly work through this so that you have some experience understanding the values of P and Q. So we have a population of flowers. There are 500 flowers in this population, and 320 of them are homozygous dominant, so they're red. 160 are heterozygous, they're pink, and 20 of them are homozygous recessive or white. So there's 500 individuals in the population. The first question asks, how many alleles? Well, if there's 500 flowers, and these flowers are diploid, they have 2 alleles per flower. So we have 100 or we have 1,000 alleles in the population. The next question is, what is the frequency of the CR allele? We have 320 individuals and each carry 2 Rs. So we can write that as 320 times 2. Here's 160 individuals. They only have 1 R. So that's 160 alleles. So all told, that ends up being 800. So we have 800 alleles. The frequency is 800 over a total number of 1,000 alleles, or 0.8. So we call this the p-value is 0.8. The frequency of the CW allele, you can work it out the same way as I did above, or if you look down here to this statement, all allele frequencies must add up to 1, you can simply write 1 minus 0.8 is 0.2, and this is Q. Okay. So note that if Mendelian processes are followed, and that is the only thing affecting inheritance, then the allele frequencies will not change from generation to generation. So as you look through the different generations, there's 4 generations here, notice that in each case, the allele frequency of R is 0.5, and the allele frequency of W is 0.5. And notice we have different genotypes represented here, but the allele frequency remains the same. So the proportions are the same. All right, so this idea of calling alleles P and Q and calculating their frequencies is just a rule of probability. So if you have an allele frequency of 0.8, it means that you have an 80% chance of pulling out, in this case, a red ball from the gene pool, or let's say the alleles in the population, and a 20% chance of pulling out one of the white balls. Okay, so here's our allele pool. Likewise, in the population, every egg has there's an 80% chance that eggs will carry the red allele, and a 20% chance of alleles of an egg carrying the white allele. This is not in a particular individual, but rather in the population as a whole. Okay, so if this is true, then we can use this to calculate the probability that 2 CR alleles will come together. So, and each egg has a 0.8 probability of carrying the R allele. Each sperm has a 0.8 probability of carrying a big R allele. And so the probability of two big R's coming together is 0.64. So this is expressed in the Hardy Weinberg equilibrium equation. And that P is the frequency of one allele, and usually this is the dominant allele. And Q is the frequency of the other allele. This is usually the recessive allele. So P squared is another way of saying AA, 2PQ, and Q squared. Notice our proportion. Okay. So when we ask about P squared, what we're really asking is the probability of a homozygous dominant individual. There are certain conditions that must be met for Hardy Weinberg equilibrium. Again, no mutations, no natural selection, no gene flow. We need a large population size, and there must be random mating. So again, we're establishing the baseline. If nothing changes, how do the alleles behave from generation to generation? And that way, if we do see some change in allele frequencies, that indicates that one of these conditions is not being met, and then you can go about trying to figure out what's influencing the population. So let's end with a sample problem here. So in this case, we have big A and little a, and this is a gene that's involved in a neurodegenerative disease. So presumably little a might give you the disease. And you have 120 individuals in this population with the following genotypes. So 16 homozygous dominant, 92 heterozygous, and 12 little a homozygous recessive. So the total number of alleles in the population, 120 times 2. The frequency of P, all right, well, we have 16 big A, so 16 times 2, and we have 92 individuals that carry the big A. This is going to be 124, and we're going to divide that by the total number of alleles in the population, which gives us, I'm cheating here by looking down, 0.52. Okay, so that's the frequency of P. The frequency of Q, well, you can go through this again, or I'm just going to take the shortcut. Okay, so now we've established the frequency of P and the frequency of Q. And this is 2PQ. So we can plug these numbers into our equation and calculate if this population is in Hardy Weinberg equilibrium, then what do we expect in the genotype? So what proportion do we expect to be big A, little a, or little a? Right, so I'm going to erase all these scrawlings here. All right, so we've plugged in our numbers that we've calculated, and what this tells us is, okay, if you have a allele frequency of 0.452, then you would expect in this population that 27%, so I figured out 27% from taking 0.52 squared, would be big A. 50% should be big A, 23% should be little a. Okay, great. Now what does this all mean? Well, this answers your question of is this population evolving? That is, what we've established here is we've said if nothing is acting on this population but normal Mendelian inheritance, then you would expect these percentages of each genotype. So now we just have to go back and plug in these numbers to see if that's the case. So 27% of 120 is 32. 50% of 120 is 60, and 23% of 120 is 28. Okay, what do we have going on here? Notice that these numbers are not in accordance. So this is what we should expect if the population's in Hardy Weinberg. This is what we actually have in our population. Therefore, our conclusion is this population is evolving. So this is how you use Hardy Weinberg equilibrium to determine if a population is evolving.